So let's talk about some of the terms that you might encounter when you're first learning about cells, especially when you get to DNA and how it's turned into protein, or when you talk about DNA as chromatin and then it's condensed into chromosomes. Also, you can replicate it, or so it can be sorted for mitosis. Well, here we have kind of a short review over all of those C words and some other terms as well, but a lot of those C terms were the crazy C's of cell division. They do pop up here and there in other places, not just when we're talking about cell division or mitosis. And before we get into each one, I'm just going to kind of briefly go through one and talk about its term and definition and what we're looking at, and then we'll go back and look at some details. So first term is chromatin. This is uncondensed DNA, but it's not just DNA, it's DNA and protein, arranged in kind of a certain order. It looks like beads on a string because you have these special histone proteins that help this uncondensed DNA have a little bit of order. But it's still unwound enough that we can read this DNA, that we can read the segments of DNA called genes. And that's what we need in order to make a protein. So in a cell nucleus for eukaryotic cells, the DNA is usually bound up in protein and exists as chromatin. Now, this is the form that is present during interphase, when the cell is carrying out its day-to-day processes, and when it's just kind of growing and developing. But then what is a chromosome? Well, a chromosome is kind of a counterpart to chromatin, but it's the condensed form. It's the condensed form of that DNA and protein that is supercoiled on itself to create bar-like bodies. And those condensed DNA and protein components, called chromosomes, they're first visible during the first phase of mitosis, which is a part of cell division. That first phase, when chromosomes become visible, is prophase. But what about a chromatid? Sounds really similar to chromatin and chromosome, so we have another C term. Well, a chromatid is a replicated or copy of a piece of DNA. So say you have chromosome A, but you have one copy. But for mitosis, we want to double the amount of DNA so that when we split the parent cell at the end, each new daughter cell has the original amount of DNA that we saw in the parent cell. Well, we have to double the DNA to do that, so you might have one chromatid, which by itself is a chromosome, but you need to copy it so that it has two chromatids. And now it looks kind of like that typical chromosome X shape. Now, when we count chromosomes, we're really just counting centromeres, which is a portion of the chromosome that links sister chromatids and actually also attaches to the microtubules that make up the spindle fibers. Now, keep in mind, we're going to come back and kind of clarify some of this information, but those are the base definitions for almost all of the C's when it comes to cell division. But there is two more, centrosome and centriole. The centrosome is a region, and that region holds two organelles of the same type called centrioles. Centrioles are responsible for creating microtubules, which when microtubules participate in mitosis or cell division, we call them spindle fibers. And the whole thing of spindle fibers being created and moving chromosomes about during mitosis is called the spindle apparatus. You can see in the image on the far right that spindle fibers are connecting to kinetochore proteins at the centromere of those duplicated sister chromatids. So again, that's a lot of C words to get used to. Let's finish up with definitions and we'll come back and make some clarifying statements. So first, spindle fibers. They're made by centrioles, but they're just microtubule structures that can pull apart genetic material. They're still microtubules, so they can also participate in moving organelles from one part of the cell to the other. They can participate in shaping the cell. They're a cytoskeletal element, but when they participate in mitosis, in cell division, they're called spindle fibers. There's also the nucleus. This is a membrane-enclosed organelle that has a nuclear envelope, a nucleolus, and a space between the nucleolus and the nuclear envelope that contains chromatin, which is DNA again organized with protein. And I kind of lied, there is one more C word, but you should be familiar with this one, cytoplasm. That is the cytosol plus organelles, or the gelatinous liquid plus cell machinery that exists between the cell membrane and the nucleus. And that's really where a lot of cell functions are carried out. That's where you can find a lot of organelles that you look at when you talk about a cell and its machinery. All right, so that's the definitions. Well, you're probably still a little confused about chromatid, chromosome, centromere, at least those. So when we're talking about chromosomes, those are just pieces of DNA, and those pieces of DNA have a central region. So here we have chromosome A and chromosome B, but they're unreplicated. There's only one chromosome A and one chromosome B. They both have one central region, and so when we count chromosomes, we count the central centromere region. So I have one central region, two central region. So that's 2 chromosomes. When I count chromatids, I'm not counting the centromere. I'm counting pieces of DNA, whether or not they're replicated or not. So if I come down here to chromatid, here on the left, I have chromosome A and chromosome B. They're both chromosomes because they have a central centromere, that dot that I can count. But they're also pieces of DNA, so they're both chromatids as well. But they're not the same, so they're considered non-sister chromatids. Well, what about what happens during S phase of interphase, when the DNA is replicated, when we double up on the amount of DNA that we will see inside of the parent cell before it enters mitosis? Well, when that occurs, chromosome A duplicates itself. Chromosome B duplicates itself. And those 2 segments, those 2 pieces of chromosome A stay bound together, and they stay bound together at the centromere, this dot. Same thing for chromosome B. It gets replicated, and those 2 chromosome Bs stay bound together at the centromere, or at the dot. Now, since there's 2 chromosome A's and they're bound together, 2 chromosome B's and they're bound together, that means I have 2 segments of DNA here and 2 segments of DNA here. And they're both the same. So these 2 A's are sister chromatids, because each piece is a chromatid. These 2 B's are sister chromatids, because each piece is a chromatid. But again, to count chromosomes, we only count the centromere. So there's not 4 chromosomes here. There's 2, just A and B. The fact that they're duplicated is shown to us when we count chromatids, because there's 4 chromatids here because the 2 chromosomes are duplicated. Whereas originally there was only 2 chromosomes and 2 chromatids, which tells us those chromosomes were not duplicated. And the centromere is just a region, like the centrosome is a region. The centromere houses kinetochore proteins, one each on the sister chromatids, and that's what these spindle fibers attach to. And then those will break down and depolymerize and pull towards opposite sides of the cell, and that's what's going to separate the sister chromatids during anaphase. So hopefully this helps a little bit with understanding all of the C terms, especially when it comes to chromatin, chromosomes, chromatid, and centromere. I hope centrosome and centriole isn't too bad. It's just a region that houses centrioles for the centrosome, and centrioles are organelles that make microtubules, which are called spindle fibers in mitosis. And cytoplasm and nucleus should be pretty easy and are covered up in the review that is posted.